Oh. All right, new setup. So today, ladies and gents, what we're going to be doing is talking about our macromolecules. So what I'm going to do is kind of introduce a little bit more background about macromolecules in general and what those really mean, like how we identify them as a particular group of molecules. And then I'm going to go into a little bit more detail about each of the individual types of molecules and what they're used for. So not only just kind of like basic functional groups and what the big picture is, but really diving into the specifics of each of the individual molecules. So you know kind of what to look for and what to be thinking about as you identify these as we continue on with our unit on our introduction to biology. All right, so let's think about a few different things. So the first thing to think about here is, right, in the 1800s, we were still learning a lot about these different molecules that you have been playing around with for the last few weeks. And essentially in the 1800s, any chemist, when they're talking about molecules, if it's a compound that came or was derived from something that was once living, we call it organic. Okay. So since they're organic, right, do you think that these types of compounds are really fundamental to living things? Like, are they necessary for those creatures to survive? And then do you think that it's possible for these compounds to exist in things that aren't living, right? So all the molecules that we're going to be dealing with, we say they're organic molecules, all right, because they are things that are found in living creatures. But when we think about organic in modern terms, we're really referring to compounds that contain carbon. Okay? So all the compounds that we're going to be looking at, if you look at the structures and as you guys have built them and researched them, you have seen that all of them contain an element, carbon, right? They have at least one atom, if not lots and lots of them. And that all of these molecules fall under this particular umbrella that is known as organic chemistry. So today we're going to be focusing on these carbon compounds that are found in these living organisms. So just to answer those questions, right, these compounds are fundamental to living things. If you do not have these four types of molecules or any of the subsequent compounds that are made up of them, chances are that living organism is not going to live very long. Right? Also, it's possible that these things could exist in non-living things. It's possible that you could have the similar compounds that find or derive themselves in other ways, or decomposing, or in other byproducts and all kinds of other things. So it's possible you're going to see these outside of non-living creatures. All right, so let's talk about carbon real fast. So since carbon as a molecule is very unique in that the sense that it's small, has a high or relatively low effective nuclear charge, right, 4 to 4, it's gonna 4 protons, 4 electrons in the outer shell. So those are going to be experiencing a +4 charge. So because of that, it's carbon, since it wants to fill that outer shell and have something that's more like helium, or not helium, sorry, more like neon, to have an outside that's filled with all of its valence shells, it can form up to 4 different bonds in order to make that happen. So carbon, for example, can form a single bond, right, joining with something else like a hydrogen, or it can form a double bond, such as with itself, right, or it can even form triple bonds. And a lot of the things that you're going to see with carbon in general is that it can bind to a lot of things. It's not really picky as long as it's going to get its valence shell. So it might join to things that have a higher electronegativity, like oxygen, for example, that has a much higher electronegativity than most other things. Oops. Right? And it might, excuse me, it might in particular, ooh, laser pointer, that's fancy. So it's possible that this particular incident or this particular material is going to bind to lots of things. So carbon is unique in that it can form lots of different bonds and it's willing to bond to a lot of different things. So that's why it ends up being very prevalent in organic compounds, because not only is it common, right, being a very light element, it's also one that's going to be present in a lot of things because of the bonds it forms. So again, carbon has the ability to form lots and lots of large and complex molecules. So it can form rings, it can form chains, it can form all kinds of crazy shapes, pyramids, all kinds of stuff, because it has that unique shape where it can form those tetrahedral shapes, right, as linear shapes, as bent shapes, as we've talked about in our previous molecule structures. So we might see something like, oops, this benzene form, where it can form this particular shape, okay, like this benzene ring, okay. So we can see all of those different patterns show up. So depending upon the types of bonds, and as you've seen and will look at in your Pierce molecules, you can have a whole bunch of different stuff happening. All right, so let's talk about macromolecules. So we've already addressed this a little bit in class, but macromolecules are big, right? That's what they mean. We'll define it particularly here in a second. So what do you think a macromolecule means? Right? And so there are 4 main groups that we are going to be looking at, which are carbohydrates, lipids, nucleic acids, and proteins. Okay. So when we're talking about a macromolecule, we're referring to a molecule that's big, right? Macromolecules essentially refers to giant molecule. And these can be made up of anywhere from 2 individual pieces to hundreds and thousands of individual pieces that are chained together. Some of the largest molecules that you have on Earth are actually just sugar molecules chained together to form giant polymers of cellulose, right? If you're talking about, say, a giant redwood tree, right, that's an entire chain all the way up through the entire length of the plant. It's just sugars that's carrying it down. So it takes millions of them to make that happen. Okay? So we've addressed that if you're going to make a macromolecule, right, you're taking these individual pieces and putting them together to make this macro or giant molecule. And that basic unit that we utilize is called a monomer. Okay? So monomers are single units that we join together to make our larger molecules, which are known as polymers. Okay? So poly means many, mono means one. So this whole process occurs through this series of chemical reactions, which is known as polymerization. So if you have something that polymerizes, that means you're putting together a lot of these smaller pieces to form a larger polymer. So to make a polymer, right, you're taking this whole bunch of tiny little things, reacting them together to give you this polymer or chain of larger molecules. All right, so let's talk about our 4 main groups. So I'll go into more detail about them, point out some major things about each of the individual groups. So you should be able to see, like if I gave you a blank copy of a molecule, to at least point out areas that would tell you like, oh, this is probably a carbohydrate because of this, or this is most likely a protein because of this. So a carbohydrate is going to be a compound that's made up of mostly carbon and hydrogen. Okay. You can also have oxygen present, and very commonly oxygen will be present, and you will see a specific ratio. So that means that for every 1 carbon, you probably have 2 hydrogens and 1 oxygen. And we'll see that a lot, specifically with carbohydrates, mostly because they form these circular rings. And just because of the nature of the structure, it's going to have this kind of 1 to 2 to 1 ratio because of the bonds of the carbon to these hydrogens. Okay. So functionally in biological systems, carbohydrates are used as mainly an energy source. For example, you as humans, right, our main energy source comes from glucose, one of the most basic monomers of carbohydrates. And we break it down through combustion reactions inside of our cells and burn it so that we get the energy from each of those individual molecules. Okay? Another big issue, or another big and useful component for carbohydrates is actually as a structural component. It's actually a building molecule. We use it a lot to build and actually make larger structures. So like I mentioned before, the wood in a tree, that's actually just a giant chain of starch, right? It's just a bunch of glucose put together to produce cellulose. It's very long, very tough, very strong structure. So your house that's built out of wood, that is built out of carbohydrate. That's why it burns very easily. That's why trees burn, is because they're made up of a lot of these molecules that will light on fire and generate plenty of energy. So an example monomer of this would be things like glucose. We've looked at glucose a couple times, but you also have things like fructose, galactose. Those are other examples of simple monomers. And then you can also have a polymer. So a big example of a polymer would be a starch, right? So like what's in potatoes, right? It's just a long chain of individual monomers together. Right. So in addition to your carbohydrates, right, we have our simple sugar, so our monomer. And functionally, monomers are pretty common for carbohydrates. They can be very energetic molecules that are used for a lot of different things. So the simple sugar molecule is known as a monosaccharide. So again, that prefix mono refers to 1. Saccharide just means sugar. So you have your galactose, your fructose, and your glucose. So you can find, say, galactose specifically in things like milk. It's common called milk sugar. Some people are unable to process it just because of the structural nature of that molecule. It still follows the same pattern of C6H12O6, that basic saccharide molecular formula, but it's structurally different. Fructose is usually found in fruits. That's why that fruct prefix that just refers to fruits. That's just fruit sugar. That's why fruits taste sweet to us. And then glucose, which is found in most other fruits. So you're going to find it in like things like a lot of your fruits and vegetables. You're going to find it in meats or things like that that contain extra glucose. So you're going to see that in quite a few different sources of energy. Table sugar. So table sugar is a simple sugar in the sense that it's small, right? It's only made up of 2 monomers, which are glucose and fructose, so sucrose, table sugar. So we call it a disaccharide. So you have 2 monomers that go together in order to build a cellular molecule. So once we get into more complex things, we're talking about polymers, and we call those a polysaccharide. So a polysaccharide is the same as a polymer carbohydrate, which is kind of the synonymous name that you might see for these polymers. So polysaccharides are really useful storage molecules because they're so large. You can have an enzyme come in, hack off individual pieces, and build a molecule that you can use and store. So humans, for example, we have a storage molecule that's known as glycogen. Glycogen is just a bunch of glucose that's put together, and it's actually stored in areas where we use a lot of energy. So for example, in your muscles, I mean, you can't see them, there's none there. But with their muscles, right, they're going to be in here, they're going to be there for ready access to energy so that as you, say, exercise or you're doing that fight or flight in the middle of some kind of terrible situation, you're going to utilize that as a source of energy. The other place you store a lot of glycogen is actually in your liver. So your liver is a storage molecule. So like you've probably heard that you need to work out for about 20 minutes before you start losing weight or burning fat. And that's because you have about 20 minutes worth of energy that's stored in your liver and in your muscles. So if you don't use all your glycogen, then you're not essentially attacking the other stored fuel sources in your body, aka fat. So you got to work out for at least 25 to 30 minutes to really start burning fat. All right, plants use a lot of different molecules, a lot of different chains of carbohydrates to store them. So starch is a huge storage molecule. Cellulose in plant structures that help supports a lot of the plants. You have other structures that are inside of plants like pectin and things that are used to store. So you're going to have polysaccharides in a lot of different places. All right, let's move on to lipids. All right, so a lipid is an interesting, at least in my mind, when I think of a as I think of our macromolecules, in that it's structurally made up of mostly carbon and hydrogen. And the thing about these guys is that you're not soluble in water. So generally, you're not going to have a lipid that's going to dissolve in water. So things like fats, oils, waxes, if you add water to them, right, they end up floating on top of the water because the fat is actually, the reason why is because of electronegativity, right? Part of it is because the actual chemical structure causes them to repel water, right? It's not attracted to the polar molecules of water. So functionally, we use lipids quite a bit for energy storage. So like, you know, it's winter time and you're getting all pudgy, right? That's because you're storing energy in the form of fat. We also use it to construct a lot of biological structures, such as membranes. So it's actually the outside of all of our cells are made up of fat. So you need fat in order to actually build your body. So low-fat diets, not necessarily the greatest thing because you're going to run out of particular lipids that you might need or the building blocks of those. Steroids and other signaling hormones are vital. So things like cholesterol, are used quite a bit into building those things. So building things like testosterone or estrogen. Without those, you wouldn't be able to signal different parts of your body properly for you to grow and function normally. So structurally, a lipid is going to be made up of two smaller components. So the first will be a glycerol. So glycerol is this right here, right? It's a chain of hydrogen and oxygens that are put together. And then you have a fatty acid. So a fatty acid is usually just a chain of hydrogens and carbons with a single oxygen double bond here that's attaching to the glycerol. So with a fatty acid, we can put them into different categories about kind of how they interact biologically with other systems. And you might have heard about these terms before. Like if you listen to the news or listen to commercials where they say like 0 unsaturated fat, right? It's referring to fats that have some kind of double bond in them. So structurally, a double bond is going to cause the molecule, essentially, instead of being a flat, straight molecule, to make it kink a little bit. And since it's kinked a little bit, it causes them to have kind of weaker intermolecular forces. So this allows them to actually move more liquid at room temperature than, say, a solid. So for example, if you have a saturated fat, that means that all of these carbons have a hydrogen attached to them, which means they have really strong intermolecular forces and that they have all of these kind of hydrogen bonds that start forming, and you actually end up with a solid at room temperature. So there's a stronger relationship with them, it takes more energy and more heat to actually split those apart into smaller pieces and to liquefy them to change their physical state. So we say that those are saturated fats. So things like lard, like coconut oil is actually a saturated fat because if it's solid at room temperature, it's saturated. Things like Crisco are saturated fats. So they're not particularly good for us because those double bonds cause them to clump and stick, and they're a lot harder for us to digest. Versus an unsaturated fat, which either it stays liquid in our body because our bodies are warmer than the ambient temperature, so greater than 98.6, greater than room temperature. So they stay liquid in our bodies and we end up flushing a lot of those out, those that we don't end up using for energy later. All right, so let's talk about nucleic acids. So nucleic acids are our next macromolecule. So they contain usually hydrogen, oxygen, carbon, nitrogen, and phosphorus. So you're going to look for kind of those 5 elements when you're looking at a nucleic acid. The other thing about nucleic acids is structurally they're going to be set up like this. You're going to see a nitrogenous base, right? So a group or rings that are going to have lots of nitrogens in them. Okay, also some oxygens and carbons. You're going to have a 5-carbon sugar. Okay, so this could be either ribose sugar, which is 5, or deoxyribose, which is missing an oxygen. So both of those will be there, so you'll see that. And then you'll see a phosphate group. So if you don't remember, phosphate is PO4. So that means that there are is a phosphorus with bound to 4 different oxygens. So structurally, they are used mainly to store and transmit information. Or sorry, functionally, they're mainly used to store and transmit information. So DNA, RNA are both examples of polymers of nucleic acids that we utilize to store that information. We often use them as well as an energy molecule. So adenosine triphosphate is an example of a nucleic acid that we actually use, because what we do is we add a bunch of phosphorus to the end of it. And by adding a bunch of phosphorus to the end of it, we can actually cleave those out and we can cause a reaction to occur, which allows us to then take the energy or the heat that's being released from that and use it in other ways. Monomer is a nucleotide. So this thing right here, this whole thing would be a nucleotide. So your nitrogenous base, your 5-carbon sugar, and your phosphate group, this would be the nucleotide. And then a polymer would be, call it nucleic acid usually when we have 2 or more of them together, or you might also see a polynucleotide. So again, a nucleotide, many of them. So we'll spend a lot more time talking about structure when we talk about DNA as a macromolecule, because it's so important and vital for our understanding of how species have changed over time. So we're going to spend quite a bit of time on the structural components of DNA. But I just want you to be able to recognize that when you're looking at it, you can see this nitrogenous base, this 5-carbon sugar, and this phosphate group. Okay. So what they'll do is they'll join through a covalent bond at the phosphate group with the sugar, and they'll form this particular backbone structure. And again, you have kind of 2 major types of polynucleotides, which are going to be ribonucleic acid, or RNA, or deoxyribonucleic acid, DNA. And we'll talk about the differences between those later. You just need to know we make DNA and RNA from nucleotides. All right, our last group that we're going to look at is protein. So protein is huge in the sense that it is not only a large group of molecules, but also proteins are really, really big, right? So proteins generally contain nitrogen, carbon, hydrogen, and oxygen. So you're missing the phosphorus that you would see in your nucleotide. And structurally, they're specific in their way. Okay, and I'll show you a little bit more about that. So functionally, proteins are used for a lot of different things. They carry out almost all of your body's functions, in that they control reaction rates in the form of enzymes. They can regulate cell processes. So if you get a signal to turn it on and off. They can form cell structural components, so like molecules outside of your cell, molecules in your cell. They're part of your structural components to building the skeleton of your cell. They're vital for all of those different areas. Transport, all kinds of stuff. Fighting disease. So a lot of your proteins will actually be signaling for whether or not something else is attaching to you. So you can check for that. And lastly, just transport. So they actually attach on to other things and carry them to other places. So the basic monomer, the building block of all proteins, is known as an amino acid. And the polymer is what we call a polypeptide, which is more than one amino acid or in a chain, or a protein, which is the chain that's then like folded on top of itself due to electrical interactions, so electrostatics essentially, of these different amino acids. So here let's look at the general structure. So again, an amino acid is a compound that contains usually an amino group, which is a nitrogen with 2 hydrogens, that bonds to some kind of carbon. And then on the other side of that, you have a carboxyl group. So you have an amino group and a carboxyl group. So the carboxyl group is a carbon with a double bond oxygen and a hydroxide group, which is what ends up reacting when we form peptide bonds. So specifically when we're forming the bonds between 2 of these monomers, amino acid plus amino acid, you're going to have what's called dehydration synthesis, and I'll talk a little bit more about that here in a second, where you actually take off a water molecule's worth of material and then stick the 2 things together to join them into a single structure. Okay? So a protein is a functional molecule that's built from 1 or more polypeptides. So again, you're adding these chains together. It can be that you just have 1 chain that folds on itself. You can have 2 chains that fold onto themselves and work together. You can have 4, you can have hundreds, you can have thousands. So it just depends on the structure of the protein. So with your amino acids, they differ from each other, right? Because they have this particular group. So if you look here, here is highlighted this R. This R is what we call the side chain, or sometimes we call it the functional group. But this particular area right here is where you're going to see the structural difference between all of your different amino acids. So structurally, they're going to have this backbone, but they're going to have this particular change. So we're not going to go through all the different functional groups because there are a whole bunch of them. But what you need to know is that there are 20 different amino acids that are found in nature. And you might have heard or might have seen on labeling for different food products that contains like 8 essential amino acids. It's because there are some amino acids that you as an individual can't make on your own. You don't have the chemical processes that allow you to produce them. So some of them we say are essential because you have to eat them in order to get them into your body. Some of them you can make on your own. So a majority of them you can make on your own, but there are 8 or 9 that you can't necessarily make by yourself. You need them from somewhere else, or you need materials to build it from something else. Right? And so because of this variation, and because of the way protein synthesis or making protein works, you can get proteins that are extremely diverse. Thousands and thousands of different types. Right? So when we're talking about the formation of a protein, like I mentioned, there are different layers that can go there. What you need to know and walk away from this slide is that proteins are forming from different pieces. So if you have your primary sequence of amino acids, we say that's your primary, right? That's just your chain, and that is like each of the individual amino acids that you're dealing with. And we'll talk about that more when we talk about protein synthesis. Secondary is when these particular chains start to fold on top of each other, and they'll form structurally more complex molecules. Tertiary is like that complete final 3D arrangement, so that's usually a functional piece of either the functional piece that is the protein that does the thing that it needs to do, or it's part of a larger protein. So you can have then the higher level or cornea level where you take the different proteins that have been folded up and then you amalgamate them together. So the example that's here, this is hemoglobin. So hemoglobin is a protein that is utilized in cellular respiration as a storage molecule for oxygen. So it actually takes oxygen in from the outside, carries it to areas of your body in order to use that oxygen, and then picks up CO2 to then be released outside. Oh, can't see anything from this slide. Let me back out. Or at least I think I can. Oh, no. All right, let me go back. So I wanted to show you these 2 different images here in a second. So let me go to animations and up here, on click, boom, on click. There we go. All right. Going on the fly. All right. Okay, so if we're linking these together, so this is not just about proteins, this is actually all of them. So the last thing I want to leave you with is just how do we build these larger molecules? How do we make them happen? And so chemically, they are chemicals. So chemically, they're going to interact with each other through chemical reactions to actually form these larger molecules. And there are specific types of reactions that occur. So remember, when we're putting things together, we have 2 smaller pieces that we join together to form a larger molecule. We have what is known as a synthesis reaction. So specifically when we're forming larger molecules in our macromolecule situations, we're doing what's called dehydration synthesis. So during this process, we're connecting 2 monomers by essentially taking away a water and joining the hydrogen from one of those to the OH group of another. That water leaves, and then the bond forms between the carbons that are present. Okay, so we could do the opposite of that, which is, remember, a decomposition reaction, where we take that larger molecule and we split it into its smaller pieces. So by taking these polymers and breaking them down, so like through digestion, for example, you're doing what's called hydrolysis. So hydro, which means water, lysis, which means to break. So if you have a hydrolysis reaction, that means you're taking the molecule and breaking it down by adding water. So you're essentially throwing water into the mix, using some kind of enzyme to split it, and then adding the hydroxide to one of the molecules and hydrogen to the other so that you're sticking them together. All right, so I'm going to leave it there. So what I would recommend you guys do is you can go back through and make sure that you understand the major components here. Take some notes because I know I talked a little fast. So you might want to slow me down here. But what we'll follow up with next is we'll talk a little bit more about kind of why these components are important as we go into actually the pieces that build your basic cell. Okay? So make sure that you review this material, that you know about each of the different macromolecules because you might see it in the future. Okay? Thanks.